Good afternoon to all the students. So, this is your Prasanna Ma'am from NEET World, and today we are going to look into two questions of your NSEJS 2022 question paper, which is the first session of this question paper. So, in today's session we are going to look into two major concepts beta. One is phytohormones, that is plant hormones, in that we are highlighting gibberellins, and second thing will be your sexual reproduction of flowering plants. In that we are going to look into the entry of pollen tube, be it through micropyle, chalazal end, or through the integuments. Based on that pollen tube entry, we are going to look into these two conceptual questions which have appeared in the year 2022. Those questions we are going to exercise with. So, without much delay, let us get started with the first question. Here in this question, the experiment on identifying this phytohormone is also given. So, in this question, it is clear that it is said that in 19th century, farmers in Japan found that some seedlings of rice become very tall. They called it as Bakane disease or mad seedling disease. All these mad plants were found to be infected by a fungus, Fusarium moniliforme. This led to the discovery of one phytohormone. So, what is this disease? First of all, what is the discovery? Usually the length of rice plants will be around 2 meters of height, but then with the effect of this fungus, drastic increase in the length of rice plant is seen on around 20 meters height. Okay. So, the reason behind this increase in the length of the plant is identified that it is because of fungal infection. That fungi is Fusarium moniliforme. Okay. And what is that phytohormone is being asked. So, if we look into the explanation, okay, the chemical which is released by this fungi is what? Gibberellin. Because of that, there is increase in the length of the plant. So, we will look into first the complete introduction, discovery, introduction and precursors, derivatives, applications and functions of gibberellins first. So, if we have to look into the concept, gibberellins are a large group of phytohormones. These are acidic in nature. Okay, gibberellic acid is acidic in nature. These are large group of phytohormones that regulate growth and development. Okay, they regulate growth and development. They are diterpenoid acids. Okay, they are diterpenoid acids and occur naturally in plants with 100 types identified, like from GA1 to GA100, of which the first identified gibberellic acid is GA3. This is the first identified gibberellic acid. The most commonly studied and widely applied form is what? Gibberellic acid 3. These hormones mainly promote what? Stem elongation, seed germination and fruit development. Okay, these are the major functions of gibberellic acid. Stem elongation or cell elongation is the most important function of gibberellic acid. Second is what? Seed germination, and third is fruit development. Discovery of gibberellic acids. In the year 1920, Japanese farmers noticed rice seedlings infected with Bakane foolish seedling disease by the fungus Fusarium moniliforme. In 1935, it is the scientist Kurosawa, he is the scientist who isolated the substance which is responsible for this infection, naming it as what? Gibberellins. Later in the year 1950, pure crystals of gibberellic acid were obtained. Pure crystals of gibberellic acid is obtained from the fungus Gibberella fujicuroi. With this marked the discovery of gibberellins, which is a plant growth regulator, that is plant growth promoter. Plant growth promoter. So, this is the basic introduction and discovery of gibberellins. Now, we learn what is the precursor and derivatives of gibberellins. Precursor of the gibberellins, like gibberellins are being synthesized primarily from acetyl CoA. Okay, They are primarily being synthesized from acetyl-CoA through this biosynthetic pathway. Gibberellins are what? Derivatives of terpenes. They are terpenoid derivatives. They are synthesized via terpenoid pathway. So, what is the precursor? Precursor of gibberellic acid is acetyl-CoA, and derivatives include what? Terpenoids. Derivative includes terpenoids. Fine. Now, so what will be the biosynthetic pathway beta? Acetyl-CoA, mevalonic acid, isopentenyl pyrophosphate. This will be the bio, and then from IPP to geranyl pyrophosphate, and then end-quarant, finally gibberellins will be synthesized. So, most important thing is you have to remember the precursor of gibberellic acid and the derivatives of gibberellic acid. Precursor is acetyl-CoA and derivatives will be terpenoids. Thus, acetyl-CoA is the starting precursor while end-quarant is the key intermediate. Now, let us look into the functions and applications of gibberellic acid. First function is what? Stem elongation is the most prominent function of gibberellic acid, which promotes the cell elongation and division of internodes. So, this stem elongation, this is used in what? Sugar canes specially. This is majorly used in sugar canes, where increase in the length of the stem will increase the yield of sugarcane crop. Yes or no? Used in sugarcane to increase the internodal length, whereby the yield of sugar is increased. Second is bolting process, whereby because of spraying of gibberellins or application of gibberellins to rosette plants like cabbage and beetroot, wherein prior to flowering when gibberellic acids are applied, post flowering when the fruit or vegetable is formed, that will increase the size and shape of the product. So, here if you see bolting, this induces stem elongation in rosette plants, wherein gibberellic acids are applied prior to the flowering, and post flowering and post fertilization, the product will be of huge size and shape. This helps in commercial production of cabbage and beet seeds. Third is seed germination. Okay. This breaks seed dormancy by inducing alpha amylase in the aileron layer and starch hydrolysis. I will tell you this with an example. Consider this as a monocot seed. In monocot seed, here embryonal axis, and this terminal is plumule, which is encapsulated by coleoptile, and bottom is radicle, which is covered by coleorhiza, and this is what? Endosperm. Okay. This is endosperm. Fine. So, during the time of seed germination, gibberellic acid will be inducing out or releasing out the production of alpha amylase. So, this alpha amylase will hydrolyze the starch and it will provide the nutrition to the embryonal axis, thereby helps in the process of seed germination. So, seed germination, this will break the dormancy by inducing alpha amylase in aileron layer or starch hydrolysis. This is used in malting process majorly. This is used in malting process in brewing industry especially, for example barley. Okay. And then next set of functions and applications. Leaf expansion, this will stimulate the enlargement of leaf cells, and then this will enhance the leafy vegetable production. This will enhance the leafy vegetable production. And flowering induction also is promoted by gibberellins. This promotes flowering in long day biennial plants under non-inductive conditions. During unfavorable conditions, this will be inducing the flower production. This is used to induce flowers, especially in the ornamental plants. This is the commercial importance. And then fruit development. Fruit development is again the major function of gibberellins, which promotes the fruit set and parthenocarpy, that is production of seedless fruits. This is used in both grapes and tomato cultivation for seedless fruits. And next function is fruit growth. This increases the fruit size and enhances cell elongation, and this is applied in apple, grape industries to improve the fruit quality and size. And then this delays senescence. Okay, this prevents seed dormancy, and this will delay the senescence as well by prolonging the shelf life period of fruits, vegetables and leaves. This will help in storage and transport by inducing or by giving out or by applying the gibberellins to the fruits and vegetables, that will help in storage and transport. And most importantly, lastly, that is enzyme induction, as the production of alpha amylase will help in starch hydrolysis, thereby providing nutrition to the developing seed and promoting seed germination. This activates hydrolytic enzymes during seed germination, which is important in research and biotechnology studies. Okay. So, these are the functions and applications. So, this thing is clear to you now beta. First thing is how gibberellic acids are being discovered. Second is the precursor of the gibberellic acid, that is acetyl-CoA. Derivatives of gibberellic acid are terpenoid derivatives. And lastly, functions and applications of gibberellic acids. Major function is stem elongation and seed germination. These are the major functions, wherein in the application process the applications to be highlighted are malting process, usage in brewing industries and malting process. This should be highlighted. Okay. And then, so once again, if we have to look into the question, so what is that phytohormone which is identified from Buchanan disease by Kurosawa, that is gibberellins, which is option D. Okay. The right answer is gibberellins, which is option D. Now, if we have to look into the reference books, okay, what are the best references for this for the learning of phytohormones? Definitely it will be photosynthesis of higher plants from your NCERT 11, and the major standard book is Theis and Ziegler, Plant Physiology. This gives you detailed information on biochemistry and its pathways, and NCERT Biology 11th, Plant Growth and Development, in which phytohormones are there, that should be learnt. Now, the second question, in most of the angiosperms, when the ovule is mature, in most of the angiosperms, the pollen germinates on the stigma. Okay, pollen germinates on the stigma, travels all across the style and ultimately enters the ovule. In the adjacent diagram, 3 possibilities of pollen tube entry are shown, which is A, B and C. Let us recall the pollen tube entry into this. First, if you have to consider this as gynoecium, okay, wherein on the stigma compatible pollen grain will come and land. Okay, compatible pollen grain will come and land on the stigma. Once the pollen grain which is compatible has landed on stigma, slowly through the germ pores where sporopollen deposition will not be there, from the germ pores slowly pollen tube will start growing or germinating all across the style. This is stigma, style and ovary. Stigma, style and ovary. So, all across the style pollen tube will come, and inside the ovary ovule is present, and in most of the angiosperms it is inverted ovule. Yes or no? So, this pollen tube will enter the ovule either through the integuments or through the chelical end or through the micropylar end. Mostly the entry is from micropylar end only. Okay. Mostly the entry is from micropylar end only. From the micropylar end, if the pollen tube is entering the ovule, we will call it as sporogamy. And if the pollen tube is entering the ovule through the chelical end, we will call it as chalizogamy. And if it is entering through the integuments, we will call it as mesogamy. So, here if you look into entry of the pollen tube, A, B and C is given, right ma. So, A it is through micropylar end, right? A is micropylar end, so it is what? Porogamy. Porogamy. And here B is through chelazal end, right? So, this will be chelazogamy. Chelazogamy. And C is through integuments, right? So, this will be mesogamy. Fine. Porogamy, chelazogamy and mesogamy. A is porogamy, B will be chelazogamy, C is mesogamy. So, option B will be the right answer. But then let us look into all the 3 modes of pollen tube entry into the ovule through comparative analysis, and we will also look into the examples in detail. So, here if you see modes of pollen tube entry into the entry in angiosperms into the ovule. First is what? Porogamy. Porogamy is through micropylar end, the natural opening at the tip of the ovule. Description: most common type of entry, pollen tube grows directly into the micropyle into the embryo sac. This is first reported by whom? Amici in the year 1824. Best examples for porogamy include lily, cotton, maize and sunflower. These are the best examples to be noted. Next, so here diagram which is representing that we have identified. Next is what? Chelazogamy, the entry of the pollen tube through the chelazal end. This is rare type of pollen tube entry, where pollen tube will bypass the micropyle and integuments. This enters the embryo sac via chelazal end. This is first reported in what? Casuarina by Trueb in the year 1891. And then examples if you see Casuarina, Juglanes, Walnut, Utrica. These are the examples. Next, last type is what? Mesogamy. This where through integuments, the entry of pollen tube through integuments or funiculus, that is the middle region of the ovule wall. Description for this: this is the intermediate pathway, pollen tube penetrates ovule via integuments or funiculus. This is less common than the porogamy. Okay. Examples include cucurbita and polygonum. These are the examples for mesogamy. So, with this we have recalled 2 concepts. One is phytohormones, that is gibberellins. Second is pollen tube entry into the ovule in angiosperms. So, what will be the reference books for this mode of question? First is NCERT biology, of course that is the major reference. Sexual reproduction of flowering plants, and then Truman's Elementary Biology volume 1 and 2 for objective practice, and the standard reference book for sexual reproduction of flowering plants. Maheshwari P, an introduction to embryology of angiosperms. This is one more standard book for reproductive biology. So, with this we have dealt with how many questions today 2 questions and 2 concepts. Coming up in our next session, let us look into 3 more concepts and the questions of NSEJS. So, this exam will not just demand the concept of yours, but then it demands so much of your objective practice. So, what we are planning is, after looking into the previous year questions, so far we have covered many questions and concepts including NSEJS 2024 paper biology, NSEJS 2023 biology concepts and questions, and now it is NSEJS 2022 conceptual questions we are practicing out. After finishing off this, we will be looking into the advanced set of questionnaire and concepts covering up the whole syllabus. So, stay tuned with this playlist, look into the all classes which is highly useful and beneficial for all the students who want to write NSEJS 2025. So, all the best students, thank you.